May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Loknetri Dr. J.D. Pawar, College of Pharmacy, Kalwan, Nasik. I welcome all of you in this new chapter, Antihistamines. In this video, that is in lecture 1, we will cover chemistry, ionization, biosynthesis of histamine, receptor of histamine, mechanism of histamine, and classification of antihistamines. The learning outcome of this topic after completion of this topic, student learning goal, chemistry, biosynthesis and interaction of histamine with their receptor, types of histamine receptors and mechanism of action of histamine. Remember classification of antihistamine with examples. So first is histamine, chemistry, ionization and nomenclature. So histamine is known as 4-5-2-amino-ethyl-imidazole. It is a chemical name of histamine, 4-5-2-amino-ethyl-imidazole. A structure of histamine composed of imidazole ring, and this one is imidazole ring, ethyl amine side chain, this one is ethyl amine side chain. The ethylene group of amino ethyl side chain are designated as alpha and beta. So methylene group means this CH2, this CH2, methylene CH2 group of amino ethyl side chain designated as alpha, and this is beta carbon. So with respect to this NH2 group, the neighboring carbon is alpha carbon, and then this one is beta carbon. The side chain attached via beta methylene group to the fourth position of imidazole. So this side chain is attached to this imidazole ring through this beta carbon or beta methylene group. The imidazole nitrogen at position 3. So here there are two nitrogen, as in case of imidazole there are two nitrogen, this one is and this one, and total there are 3 nitrogen including this nitrogen. So imidazole ring contain 2 nitrogen. The nitrogen at position 3 is designated as pros nitrogen, denoted by pi sign. It is a pi sign and it is called as pros nitrogen. So nitrogen at third position, when there is a double bond, then it is called as pros nitrogen, designated as pi sign. Similarly, nitrogen at position 1, this one, is termed as tele nitrogen, tele, designated as tau sign, like T, that is tau sign, called as tele nitrogen. So imidazole ring consists of 2 nitrogen. Nitrogen on third position is called as pros nitrogen, designated as pi sign. Nitrogen at position 1 called as tele nitrogen, that is designated as tau sign. The side chain nitrogen is designated as alpha nitrogen. This nitrogen which is attached to alpha carbon, then it is called as alpha nitrogen. So total 3 nitrogen is there: tele nitrogen, pros nitrogen, and this one is alpha nitrogen. So it is a chemistry of histamine. Now ionization of histamine. Histamine is a basic compound. The pK value of pros nitrogen is 5.80 and pK value of tele nitrogen is 9.4 and pK value of alpha nitrogen is 1.4, capable of existing as a mixture of different ionic and uncharged tautomeric species. So histamine exists as a mixture of ionic, means which contain charge, and uncharged tautomeric species. Histamine exists almost exclusively 96% as a monocationic conjugate species. Monoconjugate species means the structure which consists of one charged nitrogen. So at physiological pH, means at 7.4 pH or physiological pH, histamine 96% exist in this form, that is monocationic form. This one is monocationic form of histamine, which contain plus charge on this alpha nitrogen. Histamine at lower pH, higher percentage of dicationic species exist. So at lower pH, histamine exists in this form, that is dicationic form, where there is a two charges on this nitrogen and on this nitrogen. That is called as a dicationic species. In water or in solution form, 80% of histamine monocation exist as N-tau tautomer and 20% exist as N-pros nitrogen tautomer. So first we see here there is a ionization. So it is a structure of histamine. How draw this reaction? So it is a histamine. Then this histamine, it is called as N-tau tautomer. That is, this one is N-tau tautomer. So here it is a N-tau tautomer. So if we do tautomerism means this hydrogen shifted to this nitrogen and this pi bond shifted to here. Then it gives this structure called as N-pros tautomer. So it is a structure of histamine called as N-tau tautomer. Then tautomer, after tautomerization, that is bond shifted here, hydrogen shifted to this nitrogen. Then it is called as N-pros tautomer. Then on both sides there is a protonation. So on both sides, protonation on this alpha nitrogen. So this become NH2. Now it is NH3 plus. Similar here, NH3 plus. This compound is called as monocation. Means it contains one charge. Monocation. Then further there is a protonation on nitrogen. That is this nitrogen. So here there is a again positive charge. Similarly here there is a positive charge. So this structure consists of two charges on this nitrogen and on this nitrogen. Then it is called as a dicationic form of histamine. So histamine exists in ionic form plus uncharged form. So it is an uncharged form of histamine. It is a charged form. This one is also charged form, but it is monocationic form. It is dicationic form. This tautomer is called as N-tau tautomer, and this tautomer is called as N-pros tautomer. So histamine at physiological pH, means at 7.4 pH, histamine 96% histamine exists in this form, that is monocationic form. These two form, monocationic form. So physiological pH, histamine 96% histamine exists in this form, that is monocationic form. At lower pH, higher percentage of dicationic form. At lower pH, histamine exists in this form, that is dicationic form. In water or in solution form, 80% contain this form, monocationic, but in that 80% N teletautomer and 20% this N-pros tautomer. So in solution form, histamine 80% histamine contain N-teletautomer 20%, that is N-pros tautomer. But in crystal form it consists of primarily N this form. In crystal form major percentage is in this form. So it is called as, that is ionization of histamine. SAR studies suggest that alpha nitrogen monocationic is important for agonistic activity at histamine receptor. So this form, that is monocationic form, where this alpha nitrogen having positive charge, having the charge. So this form is required for binding with histamine receptor. And uncharged form means this uncharged, there is no charge here in this here. That is called uncharged histamine, which is contribute to diffusion across the cell membrane. So it is about the ionization of histamine. Now histamine, as we know chemically, 2,4, that is imidazolyl ethyl amine. It is an autocoid, which means it acts similarly to a local hormone near its site of synthesis. It is an endogenous substance synthesized, stored and released in mast cell, basophil and in some neuron. So most histamine in body is generated in granules in mast cell and in WBC called as basophil. Mast cells are especially numerous at site of potential injury, that is the nose, mouth and feet, internal body sulfate and blood vessels. Histamine also found in several tissues including hypothalamus, where it functions as neurotransmitter. Then biosynthesis of histamine. Histamine is synthesized in cytoplasmic granules of its principal storage cell, that is mast cell, and basophil from naturally occurring histidine, S-histidine, which is an amino acid, via catalysis of pyridoxal phosphate dependent histidine decarboxylase. So it is the enzyme histidine decarboxylase. So it is a structure of histidine. After decarboxylation, decarboxylation means removal of this carboxylic group, then it gives the histamine. So histamine is synthesized from the histidine, which is an amino acid, by the enzyme histidine decarboxylase. That is, after decarboxylation it gives histamine. So it is the biosynthesis of histamine. Now histamine receptors. Once released, the physiological effect of histamine is mediated by a specific self-receptor. Extensive pharmacological and molecular biology studies have revealed presence of at least 4 different histamine receptors in mammalian system, designated as H1, H2, H3 and H4 receptor. So histamine, there are 4 types of histamine receptor, name given H1, H2, H3 and H4, but the first 2, that is H1 and H2, is important. Most of histamine act through this H1 and H2 receptor. All histamine receptor are heptahelical transmembrane molecule and it is GPCR type of receptor. So all histamine receptor H1 to H4, it contain heptahelical membrane, that is transmembrane 1, transmembrane 2, transmembrane 3, 4, 5, 6, 7, that transduce extracellular signal via G protein to intracellular secondary messenger. These 4 histamine receptors differ in their expression, location, primary structure, signal transduction process and physiological function. So H1 receptor, so it is the main receptor. H1 receptor expression is widespread, including CNS neuron, smooth muscle of respiratory system, GIT tract, uterine tissue, endothelial cell, neutrophil, eosinophil, monocyte, T and B cell, etc. So these are the location where H1 receptor mainly present. If we see the regarding H1 receptor, this receptor is composed of 487 amino acid. So H1 receptor, regarding H1 receptor, it contains 487 amino acid. Its third transmembrane domain consists of short intracellular C-terminal of 17 amino acid and large intracellular loop of 212 amino acid. So H1 receptor is a GPCR receptor. It contains 7 transmembrane called as TM1, TM2, TM3, TM4, TM5, TM6 and TM7. But here the important point is it contains short intracellular C-terminal of 17 amino acid and there is a large intracellular loop of 212 amino acid. So it is the characteristic of H1 receptor that it contains short intracellular C-terminal and large intracellular loop. Transmembrane 3 and transmembrane 5 domain are responsible for binding of H1 ligand. So histamine generally bind to transmembrane 3 and transmembrane 5 of histamine receptor. So histamine, there is a binding site on histamine receptor which is present on transmembrane 3 and transmembrane 5. So if we see the binding of histamine with their receptor, especially H1 receptor, so here there are 3 binding sites. Acidic aspartate residue, this one, aspartic acid residue position is 107. So this one is a part of H1 receptor. So on 107 position there is a aspartic residue in transmembrane 3 appear to be responsible for binding of protonated amino group. That is a protonated amino group, that is alpha nitrogen. Protonated amino group of ethylamine side chain of histamine via ionic interaction. So it is the first binding site of histamine with their receptor, H1 receptor. So the first binding site, that is aspartate 107 of H1 receptor, bind with alpha nitrogen via the ionic bond. Then second binding site, aspargine 198. This one is aspargine 198 residue of transmembrane 5 interact with N-tele-nitrogen of imidazole. So it is a N-nitrogen tele-nitrogen. So it is interact with aspargine. Third binding site, lysine 191. Lysine 191 residue interact with N-pros nitrogen via ion dipole interaction. And here hydrogen bonding through the hydrogen bonding. So it is a 3 binding site on H1 receptor for histamine. First binding site, aspargine 107 bind with alpha nitrogen through the ionic bond. Second is aspargine 198 binding site bind with tele-nitrogen through the hydrogen bonding. Third one is lysine 191 bind with N-pros nitrogen through the ion dipole interaction. Signal transduction as H1 receptor involve activation of GQ protein that stimulate intracellular phospholipase C to hydrolysis PIP2 that is phosphoinositol diphosphate into triphosphoinositol and diacyl glycerol, that is DAG. IP3 promote intracellular calcium release, whereas DAG stimulate phospholipase A2 and thereby cyclic AMP and nitric oxide synthase production. So generally H1 receptor attached to GQ type of protein. So as we know, there are three G protein, GS, GI and GQ. So here the G protein is GQ type, which having the secondary messenger phospholipase C, and the product of phospholipase C pathway is IP3, that is inositol triphosphate, and DAG, diacyl glycerol. H1 receptor have approximately 45% homologous with muscarinic receptor M1 and M2. So histamine receptor having the structural similarity with muscarinic receptor M1 and M2. As a result of wide spread of tissue localization, H1 receptor mediate lots of physiological process, including pruritus, pain, vasodilation, hypotension, headache, bronchoconstriction, etc. So these are the main effect when histamine bind to H1 receptor. Then it produces lot of physiological process, which may be pruritus, means itching, pain, vasodilation, hypotension, bronchoconstriction, etc. But the important effect is, first one is, in case of immune response, released histamine act on H1 receptor causes increased cellular adhesion and chemotaxis. Chemotaxis means accumulation of leukocytes or WBCs, or eosinophil and neutrophil, co-stimulatory activity on B cell, blocking IgE production. It is the immunoglobulin E responsible for allergy. This process contribute to allergic inflammation associated with histamine release and are the basis for antihistamine drug therapy for treatment of allergy. So histamine play important role in allergy. So when histamine release, then it bind to H1 receptor and it produce the increase the cellular adhesion, chemotaxis of eosinophil leads to inflammation and blocking the IgE production, that is immunoglobulin E antibodies production. These all contribute to allergic inflammation associated with histamine, and it is the main basis for antihistamine drugs. Then second physiological effect, that is vasodilation and fall in blood pressure. So histamine bind to endothelial cell, causes them to contract, thus increasing vascular leak. It also stimulates synthesis and release of various vascular smooth muscle cell relaxant such as nitric oxide, resulting in blood vessel dilation. Third, that is on nasal mucosa. Histamine increase vascular permeability, causes fluid to escape from capillaries into tissue, which leads to classic symptoms of allergic reaction, that is runny nose, watery eyes. Allergen can bind to IgE, immunoglobulin E, loaded mast cell in nasal cavity mucous membrane. This can lead to three clinical responses, that is sneezing due to histamine associated sensory neural stimulation, hypersecretion from glandular tissue, and third one is nasal congestion due to vasodilation and increased capillary permeability. So it is the effect of histamine on nasal mucosa. So it is the about the H1 receptor. Then second receptor is H2 receptor. So H2 receptor, this receptor composed of 359 amino acid. The most notable difference between structure of H1 and H2 is much shorter third intracellular loop and longer C-terminal loop of H2 receptor. So as we seen in H1 receptor, in case of H1 receptor, C terminal is shorter. In case of H1 receptor, so in case of H1 receptor, there is a shorter intracellular C terminal and larger intracellular loop. But here, shorter third intracellular loop and larger C terminal loop in case of H2 receptor. Here the signal transduction of H2 receptor involve activation of GS protein. In case of H1 receptor, there is a GQ protein. But here H2 receptor, GS protein, which stimulate adenyl cyclase as a secondary messenger, promote synthesis of cyclic AMP. So in case of H2 receptor, secondary messenger is adenyl cyclase. In case of H1 receptor, secondary messenger is phospholipase C. So H2 receptor adenyl cyclase activity increases, which increase the synthesis of cyclic AMP. In some tissue, H2 receptor may link to GQ protein that stimulate phospholipase C to form IP3 and diacyl glycerol. The important effect of H2 receptor is on gastric acid release. So enterochromaffin cell located within the gastric gland of stomach release histamine that stimulate thereby parietal cell by binding H2 receptor. Stimulation of parietal cell induces uptake of carbon dioxide and water from the blood, which is then converted into carbonic acid by carbonic anhydrase enzyme inside the cytoplasm of parietal cell. The carbonic acid readily dissociate into H plus ion and bicarbonate. The H plus ion are pumped into the lumen of stomach via potassium hydrogen ATPase pump. So in short, histamine after stimulation to H2 receptor, it increases the H plus ion, that is, it increases the acidity. Therefore, H2 blocker used in treatment of acidity. So it is about the receptor of histamine. So mainly 4 receptor, but H1 and H2 is important. So in this table, receptor is given, then location is given. For example, H1 receptor, there are various locations. For example, smooth muscle, endothelial cell, nerve ending. So these are the main location of H1 receptor. Their physiological, biological effect include allergy responses, vasodilation, contraction of smooth muscle. So these are the effect. Similarly, in case of H2 receptor, location is gastric, parietal cell, cardiac muscle cell, mast cell, brain, and function is stimulation of gastric secretion. Similarly, H3 receptor, central nervous system, effect is modulating neurotransmission. Then H4 receptor, intestinal tissue, spleen. These are the location, and function is regulating immune responses. So this table in short show the location and biological effect after stimulation of H1, H2, H3 and H4 receptors. So it is about the receptors of histamine H1 and H2. Then regarding microaction, so as we know, histamine after release combined with their receptor. For example, here it combined with H1 receptor, which leads to increase the calcium level, and therefore the various effect is obtained, that is smooth muscle contraction, increased capillary permeability, vasodilation, etc. In case of H2 receptor, cyclic AMP increases by stimulation of adenyl cyclase enzyme, therefore the increase in gastric acid secretion, blood vessel vasodilation, increased capillary permeability. Similarly, H3 receptor stimulation decrease cyclic AMP, decrease histamine release, decrease secretion and vasodilation. So simple microaction, histamine after release bind to their receptor H1, H2, H3 and H4, but mainly H1 receptor and H2 receptor. H1 receptor, the secondary messenger is, that is phospholipase C due to GQ protein. In case of H2 receptor, the GS protein, which secondary messenger is adenyl cyclase, cyclic AMP, and therefore it produces their effect. So it is about the histamine. Now the main chapter, that is classification of antihistamines. So as our chapter name, antihistamine, means these are the drugs which block the histamine receptor. So generally the drug which block H1 receptor is called as antihistamine, and the drug which block H2 receptor is called as H2 blocker or H2 antagonist. So in general term, antihistamine means which block both H1 H2, but mainly H1 receptor is blocked by this antihistamine drug. So classification of antihistamine is first generation, first class is first generation, and that there are 6 classes, that is ethylene diamine derivative, example phenbenzamine, triphenamine. Then second one is ethanolamine ether, called as amino alkyl ether, example diphenhydramine and doxylamine. Third one is alkyl amines, example chlorpheniramine and phenyramines. Fourth one is piperazine derivative, example cyclizine, maclizine. Fifth one, tricyclic antihistamine, promethazine, trimeperazine. Sixth one is heptanes, example azatidines, cyproheptadine. So it is called as first generation. First generation means older drug, or first generation having the side effect, that is drowsiness. So first generation is divided into these 6 classes. Then second generation, that is called as non-sedating drugs. So it is further classified into 3 types. First one is piperidine derivative, example terfenadine, fexofenadine, estimazole. Then piperazine derivative, example cetirizine. Pyrrolidine derivative, example acrivestine. So it is a second generation called as non-sedating drug. Third one is topical antihistamine, which used topically, example leucobustine. It is an example leucobustine. Then proton pump inhibitor, example omeprazole, romeprazole, lansoprazole. And fourth one is anti-ulcer, or called as H2 blocker, or called as H2 antagonist. So as name indicate, these drugs block the H2 receptor more as compared to H1 receptor, therefore also called as H2 antagonist, example cimetidine, ranitidine, famotidine. So it is the classification of antihistamine. So generally antihistamines term is used for the drug which block H1 receptor more as compared to H2 receptor. So in that first generation and second generation is the types. Then topical second generation mean non-sedating drug, it is a newer drugs. Then topical antihistamine, example leucobustine, proton pump inhibitor, omeprazole, romeprazole, lansoprazole. And last one is H2 blocker, which block the H2 receptor more, example cimetidine, ranitidine, famotidine. So it is about the histamine and classification of antihistamine drug. In next video, that is in lecture 2, we will discuss about the SAR of first generation antihistamine, that is H1 blocker. So if you like the video, then subscribe the channel. Thank you very much.